Perfect. Okay, well, thank you so much again. I work at the University Medical Center in Groningen, and I'm also affiliated with the Netherlands Brain Bank. My research group also does other projects. But for today, I would really focus on my collaboration with the Netherlands Brain Bank, in which we aim to establish the Netherlands Neurogenetics Database. And I'm not going to show a lot of genomics data yet, because that's basically the next sort of step, but I'll allude to that a bit more later onwards. But on a big level, I always like to start this talk a bit with like the big picture. So I will give a bit of an introduction on, I think, what are the major difficulties we're facing in the diagnosis and prognosis of different brain disorders, and also why the Netherlands Brain Bank and the Netherlands Neurogenetics Database offer like unique opportunities to study disorders beyond the confines of the traditional diagnosis. And with the Netherlands Neurogenetics Database, we actually have a number of different aims. So it's very cross-diagnostic. On one hand, we look at the data from a clinical perspective by what we refer to as clinical disease trajectories. I'll talk a bit more about that. We do some predictive modeling. So we try to predict who will get which disorder or which person has which disorder based upon signs and symptoms, genetic information. We also try to stratify different diseases according to neuropathological information. So we have a lot of neuropathological information, and I will exemplify that today by focusing on the multiple sclerosis database that's related to the NND. I'll talk a bit about genetic susceptibility, and by the end, I'll only, talk very briefly on our future perspectives on how we want to aim to integrate multi-omics data types into this sort of framework. So, on a big picture, I think if you look at a random group of people, it's always very difficult to determine who will get which disorder. We know that I think almost like 50% of patients or individuals who are 85 might at some point develop Alzheimer's disease. And, a lot of other people just develop multiple sclerosis, schizophrenia, depression. I think everybody in this audience at least knows or suffers themselves from any of these sort of brain disorders. And the difficulty with all of these disorders is also that if somebody develops gets a particular diagnosis, the prognosis is incredibly difficult, right? Like even two patients with Alzheimer's disease, some of them go through the disease very fast. Other people take years. Multiple sclerosis, the exact same thing. Schizophrenia, it's the same sort of profile for each of these disorders. That it's difficult to determine who will get the diagnosis, and also very difficult to determine how the disease will progress. And I think there are a number of main obstacles that we need to overcome. So first of all, genome-wide association studies and also exome sequencing and other kinds of genetic studies have identified a lot of like variants and mutations associated with these disorders. But we still, for many of them, don't really know what they're exactly doing on a molecular and biological perspective. Second of all, for many of these disorders, the cellular and the molecular drivers of the pathophysiology are not fully understood, although with the rise of multi-omics approaches, this field is going very fast. And last, which I also think is a very important point to address, is that the relationship between the clinical manifestation of a disease and the underlying biology is really not one-to-one. For example, if we look at Alzheimer's disease patients who are diagnosed clinically for Alzheimer's disease, it turns out that if you perform a neuropathological examination, almost 30% of them actually don't have a neuropathological, the neuropathological hallmarks of Alzheimer's disease, but actually have, have other kinds of dementia, like dementia with Lewy bodies, or complex forms of dementia, mixed forms of dementia. And at the same time, people with very different manifestations can have the exact same underlying neuropathology. So there's a ton of heterogeneity. And, you see that like most studies, I also think in neurogenomics, are really focusing on the clear diseases, right? People who clearly have Alzheimer's disease, clearly have Parkinson's disease. And, most people tend to avoid the more complicated mixed forms of these pathologies. And I think that's one of the unique opportunities that we are trying to address now. And that's why we established the Netherlands Neurogenetics Database. The idea is that the Netherlands Brain Bank has obtained a ton of information from their donors. I'll talk a bit more about that in a sec. But the idea of the database is that we really want to go all the way. So we on one hand, we have genetic information. So we've obtained SNP chip data from a lot of these individuals, almost 3,000. We have very deep and extensive clinical and medical summary files, which is also really a unique feature of the Netherlands Brain Bank. When I was in California, we worked with a lot of different brain banks. And for most brain banks, if you contact them and you want to get some Alzheimer's disease tissue or Parkinson's disease tissue or any of those sort of tissues, they give you hardly any clinical information on the donors. You know, you get a chunk of tissue and, you know, you might maybe be happy if you know the diagnosis, the age and the sex, and that's pretty much it. Well, again, there's so much heterogeneity, both clinically and also temporally. And that kind of, you know, that kind of information right now is in post-mortem genomic studies pretty much neglected because most brain banks just don't collect this information. Also, neuropathological examinations are being performed by the Netherlands Brain Bank very extensively. But one of the problems is that both the clinical medical history information as well as the neuropathological examinations have all been done since 1985, and it's all done in the form of text data. So it's tremendous large amounts of text data, and you know, with the rise and the advent of new you know, text machine learning models like ChatGPT, who came out recently, but also Google BERT and many other like models that are able to process language data. Me and my research group are now using these kind of technologies, machine learning models, to convert this wealth of text data into standardized trait assessments that we can use for to learn more about the heterogeneity, both on a clinical perspective as well as a neuropathological perspective, and then integrate that information with genetics information. And in future also would like to go for like more multi-omics approaches, for which we are now processing different tissue types. So to give a bit of a background on the Netherlands Brain Bank, as I alluded to earlier, it's been established in 1985. And we've been now able to almost have, they've performed almost 5,000 autopsies. So this number is already a little bit higher than it is as is depicted here on the slide. We have 5,000 people who are registered. And one of the unique features is that our postmortem delay is really short, which is also, I think, a tremendous important variable. We know that postmortem delay really influences the quality of the material and has a tremendous amount of effect on gene expression, epigenetics data, and whatsoever. The Netherlands is a small country, highly dense, one of the most densely populated countries in the world. So we are able to, on average, have a postmortem delay of 6.5 hours. And then from each of those donors, the brain is obviously taken out of the body, dissected according to extensive protocols in a disease-specific manner, put in these freezers, and are used for international research by researchers worldwide. But as I alluded to earlier, what they also do is we have staff, medically trained staff, that basically contact all the medics, the medical hospitals, general practitioners, etc., and then try to write very extensive summaries. So we have general information, age and sex, postmortem delay. We try to write down what are the diagnoses, so what was somebody suffering from, and in which year. They write very extensive clinical history files in which they describe like different signs and symptoms and other kinds of medical problems that were occurring throughout their lives. We have information on intoxication, again, in the form of text data, like description of how much alcohol did people use, how much did they smoke. We sometimes have information on family history, like did particular diseases run in the family, yes or no. There's a ton of questionnaires like IQ, MMSE, Mini, but also like the DSM, which is particularly important for psychiatric disorders. Neuropathological examinations are being performed at multiple levels. So on the macroscopic level, there's a ton of like different brain regions that are being studied, and then we describe what there's gliosis, dystrophic neurons, and all kinds of other parameters. On the macroscopic level, so what does the brain look like in its totality? What does the circle of Willis look like? Are the ventricles enlarged? All that kind of information. And then take it together based upon the neuropathological information and the clinical information. They assign what we call is a final diagnosis, a main diagnosis. So that's a neuropathologically informed diagnosis, but there's a bit of clinical information in there as well. And this material has been, as I said, available for researchers worldwide. But most studies that have been making use of the Netherlands Brain Bank have done this in very short, small designs, right? Like 20 MS tissues and 20 controls. And the idea of the Netherlands Neurogenetics Database is that now we would really like to systematically mine the Netherlands Brain Bank, in its totality. So to come back to my overview, the first thing that we've been working on over the last few years is like to try to what we refer to as these clinical disease trajectories. And for that, we set up a workflow and a bit of a summarization of what I just showed before as well. You have the neuropathological examination, the medical records, these are also summarized into the NBB donor files. And then the first aim was that we had to identify, what are actually key signs and symptoms associated with different brain disorders. And the complexity there is that we're looking at many different kinds of brain disorders. There are databases out there specifically for, like RDoc, specifically for psychiatric disorders, for signs and symptoms specific for for Parkinsonisms, but there really wasn't a system like perfectly set up for us. So we've been playing a lot with that. And at the end, we came up with a system that contained 5 main domains. You have a psychiatric domain, a cognitive domain, sensory, autonomic domain, motoric domain, and the general domain. Within each of those domains, we identified a number of groupings. And then with each of those groupings, we identified different signs and symptoms and we defined them specifically, like agitation, restlessness, changed behavior, psychiatric admission, hallucinations, delusions, psychosis. So it's a very broad range of different signs and symptoms associated with brain disorders. And for each of them, we have been able to label a ton of different sentences. So we identified many symptoms. We sort of set up a random system and labeled approximately 10% of our cases manually, and then built multi-label classification, natural language processing methodologies to identify on a sentence per sentence level, like which sign or symptom is being described here. This is all also in a paper that's under review right now. It's also published on medRxiv if you're interested. Well, basically, we set up a workflow containing a number of different sentences. So we have like 18,000 cleaned, labeled training data sentences, of which we took a subset for training and validation, also almost 15,000. We performed a cross-fault validation design, used Optuna to optimize, to perform hyperparameter tuning, and then, basically used a number of different models. So we used like old school models like an SVC and a bag of words approach, but we also went for more modern approaches, like more like PubMed-BERT, which is like a BERT modification. This is a particular model that was developed by Google a number of years ago, and this one was specifically retrained on PubMed summaries. So it's really a model that's really uniquely adjusted for like medically trained data, let's say. We used another one, bioclinical BERT, another models like retrained, another BERT model retrained on clinical data. We also used T5 as a even more recent NLP model, and then ultimately use the test data to sort of like compare, first optimize each of them, and then in the end compare the optimal models from all of them together on the test data, identify what is the best overall model, retrained it on all labeled data, and then in the end took all sentences from all donors to predict which signs and symptoms are occurring in which sentence. And as these sentences are all, parsed according to the year, in the end, like we now have a full data set that gives us beautiful information for each of the donors on exactly which sign and symptom was occurring throughout their life. In the end, we found out that PubMed-BERT was by far what was performing the best, like relatively similar to bioclinical BERT, better than T5. And here we can see, for example, the precision and the F1 score for the PubMed-BERT. Basically 4 of them, or 1, 2, 3, 4, 5 of them didn't work as well, and the rest was really, we were quite reliably able to identify them. So we continued ultimately with 80 different signs and symptoms. And this gives us this information, and for each donor, we now have a matrix, basically a Boolean temporal matrix, as we refer to it, where you have like years, like what are the years in which people either visited their medic, their GP, or their hospital, and then which are the signs and symptoms that occurred. And then the question is, what do you do with this, right? It's like we have done this for almost, I think, 3,102 donors. So at first, we basically wanted to see, and compile these donors according to their signs and symptoms. And that's depicted here as a dot plot. And in this dot plot on the y-axis, we can see different symptoms. These are all symptoms in the psychiatric domain, like restlessness, mania, depressed mood, compulsive behavior. The color varies a bit with different groupings. And at the x-axis, we see the different diagnostic labels. So here, for example, we have control individuals. So those are people who died without having a clear diagnosis of a brain disorder. Here, for example, we see patients, everybody compiled with Alzheimer's disease. And then we see these little bars that depict the numbers. Also here, these bars depict like how many times were particular signs and symptoms observed. And then the size of the dots basically correspond to a proportion of cases within each of these diagnoses, and then the color corresponds to the mean number of observations. And I think what's most striking here is, for example, is that depressed mood is one of the most frequently observed signs and symptoms, or symptom in this case. And we see that depressed mood is very clearly very often identified in the medical summaries of donors who were diagnosed with major depression or with bipolar disorder. So that's exactly what you would expect. But you also see that depressed mood is quite common in other disorders as well, but to lesser degrees, like Parkinson's disease patients, especially those with dementia, tend to have it, and a subset of other patients as well. And mania, which is obviously a symptom that's very clearly associated with bipolar disorder, was indeed only identified by the model, predicted by the model to be happening in patients with bipolar disorder. In very few cases with either schizophrenia or patients with major forms of depression, there were a few of them, but that wasn't much. I made a mistake with these slides. I'm sorry about that. Here we have the slide that I was looking for. I should have removed those other two. My bad. But this is the full set. So I was just only showing the psychiatric symptoms, but we really have a wealth of clinical information on our donors. Again, these are just like donors with very simple diagnosis. We also have a lot of donors with weird complex forms of diagnosis. So for those, for this first visualization, we just wanted to see, does it sort of corroborate what we would expect? I already showed like how mania is very clearly defined confined to bipolar disorder individuals, depressed moods to patients with either major forms of depression and bipolar. But that was already a first sort of like pass of the validity of what we're doing. And there's so much information here. I can literally talk about this specific figure by itself already for really long periods of time. Here, for example, we see a lot of like extrapyramidal signs were associated with Parkinson's and other Parkinson's disease associated disorders, Parkinsonisms, like a tremor, rigidity, Parkinsonisms. And you see very clearly that those are confined to patients with Parkinson's disease and Parkinson's disease with dementia, as well as like other like dementia with Lewy bodies, and also some frontal like progressive supranuclear palsy, which is a frontal temporal dementia subtype or associated type, which is also, those patients also have a lot of extrapyramidal signs. So there's a lot of information that we have. And there's like, again, I can talk about this a lot, but like, just to highlight two of them, the most common ones are mobility problems, which really happen very frequently in patients with MS, as well as patients with Parkinson's disorders. So these are also like called motor diseases, where we beautifully pick up on this information. And the other one that's really like the most common symptom is dementia, or the second one, I think. So dementia is really clearly confined to Alzheimer's disease cases. Well, you would expect that. Not so much to Parkinson's disease without dementia, but very frequently to Parkinson's disease with dementia. So really like offering like proof of principle that our NLP models, that are trained on this clinical data, are beautifully able to pick up on signs and symptoms in a diagnosis specific manner. You can also see these little stars in them, which is sort of like significance over a non-parametric test, and the little circles, which is basically what we would expect based upon clinical diagnosis. And there's a massive overrepresentation of significant diagnosis, significantly overrepresented symptoms in specific disorders versus what we would expect clinically. But then, this thing was like a flattened vector, let's say, which wasn't really in any way looking at the temporal components. But we have a lot of like temporal data as well, because most of the data is parsed at the level of years. And I think that's really, quite a unique feature of what we are dealing with here. And if you look at the number of year observations, we can look at it from multiple angles, because there's so much information for each symptom. You can do this, or like combinations of them. So we really think that this is a unique resource that can be used by many different researchers in many different contexts. But if you look at dementia, for example, we see that dementia, year observations of dementia, like the year is basically the minimal resolution of time that we have for all of these disorders. We see that most of them is an Alzheimer's disease individual, Parkinson's disease with dementia, and frontotemporal dementia, and they all have kind of the same sort of distribution, while control people have basically no observations of dementia, which you would expect. We can look at like when are the dementia signs and symptoms observed on a temporal fashion. So this is age as a matter, and like there's a density plot as well as a violin plot depicting also with significance. And it's very clear that patients with frontotemporal dementia on average have the dementia on much earlier age than all of these other dementias, which is also what you would expect. But it's at the same point, if you look at these massive distributions, right, like it can be very interesting to see like why exactly are there frontotemporal dementias in this end of the distribution versus those, right? If you're interested in learning more about frontotemporal dementia, in the same way for Alzheimer's disease, why is there such a massive tail of distribution? And I think this, like with the information that we now have, we can start to address these sort of like very fundamental research questions also from a multiomics perspective, right? Are there differences in the pathophysiological development on people on this side of the distribution versus on the other side of the distribution? On another level, we can also look at like, okay, once you have a diagnosis of dementia, let's say, how long do you still have, right? That's the other part. Like how fast does it progress? Like what is your survival after dementia? And it's depicted here. And we can see that patients with Parkinson's disease, after they have a diagnosis of dementia, on average, on the median at least, is like maybe 3 years, while patients with AD or DLB or FTD still tend to have like maybe 6.5 or 7 years. I think it's 6 years. And it's depicted here as well. The significances aren't massive, but this is just one example. So with dementia, you can see that it goes in a relatively similar fashion.
Can I ask a quick question?
Yeah, sure, please.
Just about sort of the pre-diagnostic clinical history. How many years and how much information do you tend to have on these patients before they receive their diagnosis?
Yeah, for many of them, it goes back really, really far. Like we have, like, I have to say it varies per donor file, right? Like the thing is, like in the end, we just have people summarizing. So they contact the GPs, they contact the hospitals, they get these files back and they make summaries. So I think a lot of the files like go back really to like Second World War, sometimes even like 1920, 1930. For other patients, it only starts in 1980, 1990. So for some of them, it's only like 20, 30 years. While for some patients, it's literally already written down problems that they were having during birth or just after them.
And do you have any plans to sort of use that to see if anything in the medical records predicts anything, both at the diagnostic and at the, I guess, molecular post-mortem brain level?
Yeah, for sure. Like this, the thing is, we have so much information here that it's sort of like, where exactly do we want to start, right? But I think it's like these temporal components, they offer so much, opportunities. Like I'll show an example of like later on how I aim to integrate the symptomatology in the context of psychiatric disorders. I'm also doing something similar in the proposal I just wrote for like multiple sclerosis. We haven't really touched upon the dementias yet, but I think that over there it's the exact same. You can ask so many different fundamental research questions that I don't have time to write all of those proposals or do the research maybe. So that's also why I think this is a really important resource also for other people who would like to study any of these kind of disorders in more depth.
Yeah, very cool. Thanks.
Thank you. Yeah, so you can like ask very fundamental questions, right? Like for example, alpha-synucleopathies are different, like, diseases that are characterized by Lewy bodies. And we have 4 of them: Parkinson's disease, Parkinson's with dementia with Lewy bodies, as well as multiple system atrophy. And the question is kind of like, they all have different signs and symptoms. Let's say some of them are associated with dementia, others with neurocognitive, others exhibit extrapyramidal signs. But is the pathology basically the same pathology, but just striking in different brain regions? Or are there really like quantitative differences between the pathophysiological mechanisms? That's the question people are asking in the field. And we can now look at this from like a temporal perspective. Obviously, it doesn't say anything about the actual pathophysiology, but nonetheless, like it can give an indication if there are massive differences. And if you then, for example, look at bradykinesia, which is one of those like extrapyramidal signs, we very clearly see that bradykinesia is much earlier in patients with MSA compared to any of those other types of alpha-synucleinopathies, suggesting that MSA indeed has like uniquely qualitative difference to it. But for example, PD and PDD, so Parkinson's disease with and without dementia, they're extremely similar in their survival after the first observation of bradykinesia, suggesting that those are probably the same pathophysiological mechanisms. But dementia with Lewy body and progressive supranuclear palsy, as well as MSA, really have a very different profile from those two. So this is, indication of the fact that, probably there are different neuropathological processes underlying a subset of those disorders. So that's an example of what we can do with this data set. Another example like that we want to continue upon is also the predictive modeling. So we have these signs and symptoms now. Can we then build like AI models to, predict who will get which specific diagnosis? And as I said, Alzheimer's disease tends to go wrong like 30% of the cases. Can our models do better? And how about more rare disorders? So for that, we implemented a GRU-D model, which was published, I think, in 2017. For us, we looked at like 2,150 donors, again, performed a cross-fault validation, and then ultimately, optimized and tested the model on, a subset of the data. This figure is not the best quality, unfortunately. I should have maybe done it again. But, it nicely shows that like control individuals are, appropriately, correctly assigned a control label, like 0.89, 89, proportional 0.89, so 89%, let's say. Alzheimer's disease are correctly diagnosed 0.91% by our model. We see that PD and PDD really don't go well, but again, those are probably just the same disorder anyway, so it's not that surprising that the model has a little bit more difficulty with those two. Vascular dementia is really difficult. We see a lot of like misdiagnosis with Alzheimer's disease. I think this is also known in clinic as well, so our model performs at this point just as poorly as the clinicians are doing. Frontal temporal dementia goes well often, but also a large subset is, misdiagnosed as Alzheimer's disease. MS does really well, bipolar does really well, and then some of these other disorders. So it's not perfect yet. But what we're doing is with every new data type, so now we are working on the genetics data, for example, there's the next step. We would like to integrate polygenic risk scores here and see like, okay, how do those do? As a next step, we can, instead of using these main diagnoses, we can also use the actual neuropathological values. So the models can become increasingly more sophisticated, and then hopefully by adding extra levels of sophistication, the models will become better and better at these sort of predictions. At least that's my hope. But that's another part of what we're doing. At the same time, as I alluded to in the beginning, or I mentioned in the beginning, is that for a lot of these disorders, there's a lot of heterogeneity, right? Like, is Alzheimer's disease just one disorder, or the multiple types of Alzheimer's disease? Right? Those are the kinds of questions that everybody's like asking. And these are difficult questions. I'm not saying we have all the answers here, but those are things that we're trying to look at. One of my students over the last years really worked on specifically focusing on neuropathological data associated with multiple sclerosis. So we have 220 donors, I think, with MS that were very extensively also neuropathologically characterized. And then we've been using like different dimensionality reduction approaches to sort of like, can we find subclusters of MS, and then, how do those, are those just like, preserved across different data types, let's say. So if we input neuropathological data, do we then also see that they have different clinical features or other kinds of neuropathological data, or maybe associated with different genetic risk scores? This is also under review right now. So we had 228 brain donors with multiple sclerosis that were very deeply neuropathologically examined. We have data associated with white matter lesions, microglia morphology, how many lesions, how many reactive sites, did they have nodules, coughing, cortical lesions. We can talk about each of those, but there's just, you have to imagine just a lot of different neuropathological parameters. And in the end, we just performed like a factor analysis of the mixed data and, performed clustering. And then in the end, we wound up with, I think, 4 clusters. And I used other data types as sort of orthogonal validation to determine, are the clusters really biologically, medically meaningful? 5 dimensions were included, and you can see how the dimensions were loading on the different neuropathological parameters. Like dimension 2 is really clearly positively contributing to active ramified microglia. Component number 1 is mostly driven by inactive and remyelinated lesions. Active rounded are driven by a really massive influence on factor number 4. And these are just like if you plot the dimension 1 and 2, and you see how the clusters are, they're kind of overlaying, but there are like quantitative differences as well. So it's a question, is it really like qualitative differences or quantitative differences? We're not sure yet, but there are definitely differences. That's quite interesting. And then we relate those back to the other data types. For example, are there differences between sex? Well, not really. Then we look at MS phenotypes, right? People in the field of MS like to subcharacterize patients or MS patients according to relapse remitting, secondary progressive, and primary progressive. Although I hear more and more MS neurologists saying that that system is really not a particularly well-defined system, and there's a lot of flaws with it. But, that's also kind of what we see. It wasn't really like clear that like one of those subtypes is associated with our neuropathological clusters. But then if you look, for example, at age, we do see that cluster number 4 especially is much earlier, and like when the neuropathology strikes, MS starts to strike, the duration of the disease is much shorter too. So it's interesting, that clinical differences. And then if you look back also in our NND clinical data that I just showed before, that was like based upon the NLP models, we see a very similar pattern. So here we again see the 4 clusters in control, and it's very clear that, for example, for decline, deteriorated health, fatigue, mobility problems, positive and negative symptoms, muscular weakness, a lot of like symptoms that are really crucially important for multiple sclerosis, that they're really much earlier, especially in cluster number 4, and then cluster number 1 and 2 are always later ones, it seems. So also that corroborates the idea that there might be differences clinically. We also saw more neuropathological data, but I'm not going to show that for today because otherwise I'm going to get stuck on it too long, and I only have a few more minutes. The next level that we're interested in is genetic susceptibility. So we obtained polygenic risk scores for almost 2,800 donors, and pretty much all of the more recent donors, I think from the last 20 years or so, 2005 and afterwards or so. And the GSA chips contain approximately 600,000 variants, which we imputed genome-wide. And now we've been calculating polygenic risk scores, which is the sum of the effect sizes of independent variants for a lot of different GWAS data sets. We'll look at Alzheimer's disease, Parkinson's disease, but also schizophrenia, bipolar disorder, and many other disorders. At first, we used the traditional PRS calculations, but we recently switched to quick PRS, which is basically a variant of mega PRS developed by Doug Speed and others. And I can just like briefly show you a few examples, but here we calculated the GWAS heritability or GWAS AD PRS score. And then we ranked all of the individuals and then compared them between each other, like are there any differences? And what you can see for an Alzheimer's disease polygenic risk score is that indeed in Alzheimer's disease patients, it's significantly higher than in many of these other disorders. Okay, that's not so surprising, obviously, but still nice to find. But what I find more interesting here is that we also have a lot of people with mixed forms of dementia. So we have patients with Alzheimer's disease and dementia with Lewy body. And those also tend to have very high Alzheimer's disease PRS scores. We have patients with Alzheimer's disease and chondrophilic angiopathy, which is another neuropathological hallmark, also very high, as well as vascular encephalopathy. So those are really on the high end of the distribution. But then what is a really interesting observation, I think, is, for example, that patients with mixed Parkinson's disease, Alzheimer's disease, actually have a much, much lower PRS score for Alzheimer's disease. So that's one of the things we need to look into, right? But how is that possible? I would have assumed that somebody who has both PD and AD has a heritability, a high polygenic risk score for both of those disorders. But one way or the other, that doesn't seem to be the case. Which by itself is really, really interesting. And as I said, we want to include these PRS scores also will start relating to other clinical as well as neuropathological features as well. You can imagine that maybe patients with a higher PRS score for Alzheimer's disease also have AD at an earlier age, or maybe have slightly different symptoms associated with them. There's all kinds of research questions that we can now start to address by integrating the genetic data on one hand, as on the clinical and the neuropathological data on the other hand. Another example, which I think is kind of interesting, was like the Parkinson's disease polygenic risk score. So you see that Parkinson's disease also on the higher end of the distribution, and we don't have as many Parkinson's disease cases, but it seems to be higher than at least some of those disorders. And Parkinson's disease or dementia is even more, but as again, those are probably just the same disorders, and the labels are really driven only by the dementia, yes or no. But what I think is really, really interesting is that PSP, progressive supranuclear palsy, which is a frontal temporal dementia subtype, actually tends to have very high PRS score for Parkinson's disease. And it's especially interesting given the fact that PSP is also associated with a lot of extrapyramidal signs. So clinically, it looks like Parkinson's disease. They have a lot of the parameters associated with Parkinson's disease, like Parkinsonisms and ataxias and all that kind of thing. And it's really interesting to see that they also might have a genetic component associated with Parkinson's disease as well. And in that sense, we could even argue, like, is PSP really a frontal temporal dementia, or is it actually a Parkinsonism related disorder? I don't know. I don't know if there's any alpha-synucleinopathy in these PSP cases, but that's definitely something I would be interested in to look into in more depth. So that's the genetic susceptibility part. That's what we're working on right now. And then the last part, which I also think is really an essential point, is that in neurogenomics, as I said in the beginning, like right now, a lot of the studies that are ongoing focus on disease cases versus control cases, right? You focus on Alzheimer's disease and non-dementia associated controls and try to see what are the neuropathological features associated with them, gene expression profiles and open chromatin and that whole thing. But, like equally important is like, why do some patients with multiple sclerosis develop fatigue? Well, other ones don't. Some of them develop depression, other ones don't. There's so many, like the heterogeneity I spoke about in the beginning is so prevalent in so many of these disorders, and especially I think in psychiatric disorders, right? Like right now, the diagnostic system, the DSM, is really focusing on clinical signs and symptoms, right? Like you have 5 out of these list of 10 signs and symptoms, and then you get schizophrenia label, and you have another set, and then it's bipolar, and another set, and then it's depression. But like more and more studies are indicating that most likely these studies are on sort of axis, right? And they're kind of related to each other. And with the current situation that for most brain banks, we don't know anything about the clinical manifestation, and everybody has the same problem, right? They do multiomics profiling on schizophrenia cases and bipolar cases, and they compare them and they say like, well, it's largely overlapping. Yeah, well, then what does it mean, right? Like it's not really unique for these disorders. But the cool thing is now that we, for example, have a lot of quantifications of these specific, the underlying symptoms. Like the symptoms are ultimately what drive, what determine if people get a particular diagnosis. So why even work with diagnostic labels? Why not use quantification of the symptoms? Right, like hallucination, here we see different distributions. For example, you can look at depressed moods, the one I spoke about earlier, and you can see that, well, most cases with control and schizophrenia do not have depressed mood. Some people with more controls also have a little bit, and you do see that a small proportion of cases with schizophrenia actually are quite well suffering from depressed mood. Right, and so instead of looking at depressed, at the diagnostic label, maybe we should try to identify what are the features with the depressed mood symptom instead of the diagnostic labels. And the same for hallucinations. There are patients who have bipolar as well as schizophrenia. Both of them can manifest hallucinations at some point in time. Anxiety is another one, right? It's another sort of feature. So we can either look at the individual signs and symptoms, or we can look also, like we're also performing dimensionality reduction approaches to try to see if you can make some conglomerates that we can more find an axis of hallucination, psychosis, more of a depressed mood, anxiety axis, and maybe there's a third like fatigue axis or something, but that's still ongoing. Well, we have written a number of proposals. I just received funding from the Brain and Behavior Research Foundation to now perform multiomics or gene expression profiling. We're collaborating with Guus Smit and Sabine Spijker in Amsterdam, who want to do proteomics profiling. I also wrote an ERC That's on the reserve list. So hopefully I'm going to make it to the full list, and then I can do many more other kinds of multi-omics profiling approaches to really in-depth start studying schizophrenia, bipolar, and major depression, not from the angle of the diagnosis, but from the angle of the underlying symptomatology. And then hopefully we can identify drivers of the pathophysiology that are more independent for really clear specific manifestations of specific signs and symptoms. And I think my time is pretty much up, but I think somebody was asking a question.
Yeah, there is a question in the chat from Yuri Meisen. He's asking, is your methodology of looking at symptoms rather than diagnostic labels in line with the NIH project RDOC? I don't know what that is. Maybe you do.
RDOC. Yeah, no, I think RDOC, we looked into RDOC in the beginning when we were defining the signs and symptoms. The RDOC, I think, was mostly focusing on the psychiatric disorders, if I'm not mistaken. So we use it at first, but like we wanted to set up a sign and symptom system that also works well for like dementias, that works for like ALS, for motor disorders and MS. And so, you know, it's sort of the same kind of philosophy, let's say, but our system isn't exactly in line with RDOC. But I think the philosophy behind it is though. So really good question. Yeah. So to conclude, we are established the Netherlands Neurogenetics Database, which I think is quite a unique resource to support studies into the relationship between clinical manifestation, genetic susceptibility, and the neuropathology of a wide range of CNS disorders. I think that our resource can facilitate both like diagnosis, prognosis of different brain disorders. On one hand, epidemiological studies, and at the other hand, you know, also try to see ourselves, and other research might also be interested in, you know, more of artificial intelligence approaches. I think it really now allows us to start drilling into genomic features associated with specific signs and symptoms. Maybe also detection of disease endophenotypes, right? There might be a lot of heterogeneity between Alzheimer's patients, a lot of heterogeneity with multiple sclerosis individuals. And I think with the data that we're now trying to collect and build together, like we can hopefully drill deeper into those different endophenotypical changes. And I think that the NNA hopefully is going to go online maybe in March or in April this year. There's just going to be version number 1, so that's only going to be the data of the clinical part. But our aim is to, like, you know, then we're going to drill into the genetics data, and that's going to be added. Then we're going to drill into the neuropathology data, that's going to be added. So I hope that we can really build a resource that gradually expands, and that with every paper that we get, like, we'll build on top of it. I'm really happy, you know, that I have a really great group of collaborators. My collaborators, the Netherlands Institute for Neuroscience, the Netherlands Brain Bank, have been really instrumental. Minke Groot, Megan Herbert, under supervision of Annemieke Rozenmuller and Inge Huitinga. In my own group, Nienke Mekkes, my PhD students, have done a lot of work. Eric Hoekstra, technician. Alice de Boer has done the work on the MS, which I'm also really proud of. Everything is done also in collaboration with Bart Egge. So if you're interested in like accessing some of the data or see potential possibilities for collaboration, let me know. This is my email address, and obviously you can also follow me on Twitter. So again, I want to thank everybody, you know, for inviting me to speak here today. It's been a pleasure. And if there are any questions, I'm happy to address those. So thank you.